Yeah, I think I'm now online again. It always takes a couple of seconds for the stream to show up. Yes, everything seems to be working. I can also hear the sound. At least I can try to hear myself a little bit here. So everything seems to be okay. I want to welcome all of you again to another Saturday microscopy live stream. I see that many of you already started to write comments in the chat. Yeah, welcome back. I say welcome back also because last week, as some of you might have already noticed, I did a pre-recorded live stream. So basically, I was on holiday last week, and still did not want to skip a live stream. So I pre-recorded one that is a little bit shorter. And today I'm actually really live again. However, there is going to be a little change in format. It has been a request. Actually, I got a comment in the last week's video that somebody actually really liked the fact that I did not always interrupt myself answering chat questions. It basically, what I would like to therefore do today is the following. In the first hour or so, I would like to simply do now a live stream. I'm going to show you now my new microtome that I have over here. And I would like to ask you, please, if you want to, of course, please do post questions and comments in the chat. And then after 1 hour, what I will do is I will answer all of those questions collectively. Okay, so I want to keep the first part a little bit more compact. And then those people who are then interested in having some of the answers, then I will cover them in the second part of this live stream today. So please do not be surprised if I do not respond to your questions right away, but please do feel free to write comments and questions, okay? And after approximately 1 hour, I will then go through the answers here, okay? Yeah, again, I'm happy to see so many of you again all around the world. Greetings to everyone. I'm not going to read out the different countries now. It has become a tradition a little bit to always say from where you're from. This little live stream here kind of uniting a little bit the whole world, the whole world united by this same common hobby, microscopy. But today's live stream is going to be a slightly bit more technical. Of course, I will put some stuff also under the microscope, of course, but I do not only want to show you my new microtome that I have over here. It's a little bit unusual looking, okay, so we'll talk a little bit about this, but I also wanted to show you this interesting little adapter ring here for connecting a DSLR camera to a microscope. I just discovered this one here, and really that's all you need. I cannot believe this. I'm going to make a separate video of this for the channel, but I'm kind of excited about this, and I've gotten so many emails on how to connect the DSLR camera. So this is might be. Yeah, there is a small caveat, but it might be indeed a very low-cost solution as well. So I'm going to talk about this as well. So what I've got over here is 3 microscope knives. I will talk about those as well. And here I've got a new microtome. First of all, important to say, not sponsored the video. I actually bought this. Yeah, I put the link into the description. No, you cannot. I did not find it on Amazon. Okay, I bought this from a UK company from the United Kingdom called Brunel Microscopes, and they've got all sorts of microscope equipment. I already bought several times something from them, and I like this thing. Really, it's a little bit unique looking. I will talk about this a little bit in a second, and how it's similar and different to the other microtomes that I have. And if you're completely new to this whole topic, because sometimes there are also beginners watching, microtomes are used to make slides. I just want to show you. I don't know. I've got over here a slide here. This is a carrot, and I stained it in blue to make very, very thin cross sections, and this one specifically of plant material. Okay, so that's basically the one that I've been using. You can see that there are already some, I've been already using it today quite a bit. So there's some slight scratches on here, but that's perfectly fine. I want to show you, first of all, my 2 other microtomes that I've been using, and the advantages and disadvantages, okay? So this one and some of you already know them because you've been watching the videos and live streams already before. That's the one that I've also been using. And you see that over here, there's a clamp in here, okay? You can use this screw here to clamp the specimen, and by rotating this bottom part, you're kind of advancing the specimen up, and then you can use a knife, a very sharp knife, yeah, like this one over here, and then you kind of slice across it. I'm going to show you how to do that, right? Now, the one that I have over here does not have a clamp, and it still works quite well. The one with a clamp has advantages and disadvantages. Just a short intro here, because if you put a specimen in here, because then it's only supported by the clamp, and it's a little bit wobbly. Okay, so there is some flexibility here, and this basically can result in sometimes the cut not being completely even. Okay, so that's a little bit the disadvantage here. The advantage is that it's held quite tightly in place. Okay, so yeah, you see that it's fairly cheaply made. It does its job, yeah, but you see, yeah. There are tolerances here, and especially when you're dealing with very thin cross sections, you do not want to have a lot of tolerances. But it works. Okay. So another one that I have is the one over here. Okay, and you see there's a little piston in here as well, and this one just is dropped in here. Okay, and you see there's no clamp, and then the specimen is placed in here, right? And then it's also advanced, yeah, using the wheel down here, yeah, and it can also be connected to the table using this clamp over here. I added a little bit of cardboard around here to prevent this from being scratched by the screw over here on the side. Yeah, so it also works. I've also been using this one, and I've also made videos before, so some of you might also know this one over here. Okay, and I did find that sometimes the diameter might be, I mean, depends really a lot on the size of the specimens, but I thought it was kind of a little bit small, the diameter. And then I quite accidentally discovered this one over here, and it was fairly affordable. It was only around 40 UK pounds, which is really, yeah, the others were more expensive. There are some delivery costs, of course, but when it arrived, I kind of like the unique shape a little bit, but then I just want to show you, it can be directly mounted to a table using this clamp, but then I just want to show you something here, because that's the wheel that you have to turn, okay, to advance the specimen upwards. And you see in this case the piston in here is fixed in place. It does not fall out. But look, I just want to show you something. If you rotate this, yeah, you actually see this? Yeah, it actually locks in place stepwise. Yeah, and there are zero tolerances, so nothing wobbly at all. Yeah, so this was actually, it makes a very, very solid impression. Okay, so I've been kind of experimenting with this now for today, and it did also come with an instruction, and they actually sent it to me over email, so it's nice to have actually someone to talk to. Yeah, and this is what they sent me. So they sent me a PDF with some instructions here. Okay, very straightforward and direct instructions, and the important thing is what they say is that whatever you put in here, it should be kind of squeezed in a little bit. I like the way that they've written it here. Look, I just want to show this to you. It's because they're being so honest here. The bundle, what you put in here, should be a little larger than the well. That's the well here, all right? So that it can be forced tightly into the well. Yes, yeah, and the procedure is messy. Yeah, so you really got to squeeze it in so that it's really stable in there, and then you can keep on advancing it upwards, and then you can make very thin cross sections. Okay, so I think one of the important things is that these microtomes, these handheld microtomes, of course, cannot compete with the microtomes, the big commercial microtomes that are used for making commercial specimens. Cannot compete, obviously. But honestly, they kind of do their job, and you have to do a little bit trial and error, because if you make several sections, then some of them are going to be better than others, obviously. Okay, so for hobby purposes, I think and for casual observation, definitely something to have a look at. Okay, so yeah, again, if you just joined in today, I'm not going to be answering the questions during the live stream. Well, yes, I will be answering the questions during the live stream, but I will make a separate after 1 hour approximately. I will make a separate Q&A session, and I would like to ask you please to type in your questions before that time, because I will actually get this printed out today, and then I will go through the questions in one block. Okay, so that's basically the thing here. Okay, so what I would like to show you now is I'm still also learning how to use this device. So if you have any suggestions on how to improve it, also please comment on that. So what I'm going to do now is I'm going to try to make a fit the carrot. Okay, the carrot will also be our specimen holder. Will all be clear, and I will kind of show you, first the wrong way. Okay, because I want to show you the problem that some of you might actually encounter and that I have encountered as well. Okay, so first of all, notice that the other microtome here, and that's a little bit a nice thing. This one over here came with a little round tube here, which exactly has the same size. So I can actually kind of punch out the carrot. Yeah, so this is actually quite a nice little convenient tool. Unfortunately, this one doesn't come with one, so I'm going to buy one from a hardware shop. I'm going to buy this hole punch. It's about 2.5 centimeters, around an inch across. So what I have to do is I have to cut the carrot, kind of estimating it. And what I'm going to do first is I'm going to show you the problem if you do it wrong. Okay, so and then I will simply use the carrot over here. Okay, here now you can see it. I'm going to get a piece of carrot, and what I'm going to do now is I'm going to cut the specimen too small. Okay, so let's try this. Okay, it's still too big. I'm going to cut it too small. Okay, like this, and this will be now our specimen holder, and I would like to microtome this leaf over here. Okay, so what I'm going to do now is I'm going to cut a piece of the leaf. So that's the one that I'll be using. And I'm going to now make an incision, a cut into the carrot. Okay. And I'm going to put this in here as a holder. And then I drop it in here. So what I'm going to do now is I'm going to move this camera. I would like to show you now the edge of the table. Okay, I hope that this actually works. Okay, and here it is. I'm going to tighten it, and maybe I'm going to just move this over a little bit like this. You have to be careful that the camera doesn't fall over. Let's do it like this. I see that the USB cable could be a little bit longer as well. Okay, here you see my feet. Okay, so here we go. I think it's easier for me if I move this over like this. Okay, here we go. So you see the carrot now on top, and here inside you see the leaf. And microphone is still close by, and I'm going to now use a knife. I'm going to use this one here. That's the old one. Okay, it's a little bit corroded as well. And this is a problem. If it's out so much, I'm going to carefully cut it off. Okay, and then I'm going to advance the bottom one by rotating it a little bit. And I'm going to now show you the problem, okay? Because maybe some of you will have a similar problem. I'm going to exaggerate it a little bit. I'm going to tighten it here. Look what happens if you cut it, okay? I know I'm exaggerating right now, but essentially what you get is you're getting some kind of a wedge, okay? While you're cutting it, it lifts out the carrot, right? And that is basically not a solution. This is the reason why they're actually saying you have to kind of firmly press it into the well so that the specimen or the carrot is held tightly, okay? And this kind of also explains a little bit the advantage of those microtomes that have a clamp, okay? Like the one over here. However, there is another problem with this here, which I also would like to show you. Yeah? I'm going to prepare again a carrot, okay? You're not seeing this right now. Okay, so this is the carrot, okay, and you know what, I'm just gonna move this back for right now, okay, and when you put it in here, and when you tighten the clamp, okay, and when you cut across, I always have to be careful, glass surface is on here, okay, now even though it's now held in place, okay, that's the nice thing, but if you advance it out a little bit, you see that essentially, because it does not touch the edge here on the top, there is a little bit, let me use the tweezers here, a little bit of a space here, right? And I'm not talking about this big space over here. Okay, now it's in focus. I'm actually talking about this tiny space here between the glass. Yeah, this basically means it's a little bit wobbly. Okay, So there is no support here on the side. So even if it were to fill out the inner part in here, it's still not quite touching the edge of the glass. So there is on the very top millimeter or two, there is no support. Okay, so this also adds a little bit of tolerance here, and this is one of the reasons why I kind of like this one over here a lot, because you see the support goes all the way to the top. Yeah, so the carrot will actually touch the rim over here as well. Yeah, and so this is actually one of the things why I was actually able to get slightly better cuts than with the other microtomes. Yeah? But again, it also depends a lot on the knife. It depends a lot on other things as well. Yeah? So what I'm going to do now is I'm going to try to make a few microtome cuts now using. I have to move this over again. Okay, using this microtome over here. Okay, and if anyone from the company is watching this, and maybe I'm just going to send them also a link to this video. I really like the microtome, but there could be one tiny little thing that would really enhance it if you were also able to, kind of include some kind of a tool like this that would actually help you punch out the carrots. That would be quite nice, but if not, it's also fine. I've also managed to do it without. Okay, so it only takes a little bit more time. I'm going to remove all of those carrots here, and I'm going to try to do it like this, and I have to kind of see if it fits. You see, see it on the bottom here. It's too thin. So let's try different. Let's try it a little bit bigger. So, and now I have to kind of cut it into shape a little bit, and I think I'm going to increase the height of the camera now again. This, by the way, in case you were wondering, it's a document camera. It's actually one for taking pictures of books and so on. And I will now have to kind of try to estimate the size. Maybe I don't have a ruler with me now, unfortunately. But I'm going to estimate the size now to be like this. Yeah, this kind of seems to be working. Okay. And over here as well. I know it's kind of difficult to show you now. Okay, we're slowly getting there. Okay. So I'm going to cut off. I know this is not entirely round, and it doesn't touch all of the edges quite well, but I hope you get the idea. Okay, slowly we're getting there. Okay, here we go. Okay, yeah, see, it seems to fit in here. Yeah, there is no tolerance, no wobbling. I know that there are some gaps here on the side as well still, okay? But maybe this works, okay? So next thing is I'm going to just carefully cut off the excess so that I can again make a little cut to hold the leaf. Yeah, and as you see, if you start cutting it, you're kind of lifting it out as well. I'm going to use this here. So here we go. Okay, and another cut. Usually I just cut it halfway so that it's held together on the bottom. Yeah, it's just for added stability. Another leaf. Okay. And let's try to fit it in here somehow. Okay. That's fine. I'm going to remove this later on. It goes in here. Okay. And I'm going to clamp it again to the table. So, yes, I see that there are people asking, is this a microtome? So maybe you just joined in the chat. Or the live stream. Yes, I'm just showing you my new microtome, which just arrived a couple of days ago, and which I kind of like a lot. Again, each device has its own advantages and disadvantages, but this one actually has, I would say, more advantages than disadvantages. How do I do this? Here we go. Here we go. The camera is tipping over. I have to add a little bit of weight. Just going to add my other microtome here. Okay, and now I'm going to turn the bottom. Oh, wrong direction. Okay, you have to turn it. I have to turn it clockwise, and I kind of feel it advancing out. Okay, and now I'm going to use a knife. And you know what? Because this knife here is a little bit bad and corroded already, I'm going to use the fresh knife that came with this microtome. Okay, looks all nice. Okay, so yeah, that's the first cut. Okay, just make sure that the knife touches both edges. So you should not be cutting it like this. Yeah, because you need support on both sides. I remove this. I advance it a little bit. Okay, and I try to make another cut. Okay, and you can actually see over here that there is, maybe you don't see it, but this one over here, that is the leaf. You know, maybe you need some kind of no, here it is. Okay. So what do you do with this? We're just going to have a look directly at it. Even though it is quite thin, I found out that it's in many cases still a few cell layers too thick. And because it's green, because of the chlorophyll, you still do not see the cells quite well. Okay, so I'm going to show you how you can actually remove the chlorophyll. And we can, if you want, we can also look at the carrot. Huh? Okay, and you see sometimes it's just slides over it. That's more of an issue of the knife. Okay, yeah, and here that's another one. Here it is. Okay, here's another one. So I put it also on here. Yeah, and you can keep on doing that. Yeah, if it's really tight, then you're actually not lifting it out either. Yeah. Sometimes it's a little too thick, and sometimes you're kind of gliding over it. Yeah. And maybe I need a little bit more practice in actually finding the right angle. Yeah. As so often, a question of trial and error. Okay. And I've got now 3 cross sections here of the leaf, and you know what? Just for the fun of it, I'm going to also add, one of those carrots here as well. Let me quickly cut off a small piece. So here are the 3 little, and then let's also put the carrot, small section of the carrot as well. Again, with those commercial big rotating microtomes and with the sample properly embedded in paraffin wax and so on, of course it's an entirely different issue. Okay, but I'm going to show this to you now, and in my mess that I have here, I don't even know where I put my pipette. I don't know. Here it is. Okay, here we go. And I'm just going to add a little bit of water, because those specimens, they like to dry out quickly. And I don't know if they're actually on the corner now, on the edge, or if this is a top-down view. I don't see that. I've already got bad eyesight as well. Okay, so I'm not even going to bother cleaning this here, and let's give it a try. Okay, so where is the scope view? The scope view here is okay, and let's have a look how this looks like, and I will tell you right away, it's probably going to be too dark. Okay, so that is the carrot. But we're able to see, let me open this a little bit, but we're still able to see some bubbles in the individual cells, and usually at the edges, we're able to then see it a little bit better. So I will also remove so this is how the carrot looks like. Okay, let's go up a little bit with the magnification. Don't forget the specimens are quite stained as well. So this seems to be a fairly thick specimen. But if you focus, so these are several cell layers, not the best cut. Okay, I've also this is actually not really good. This is too thick. A little bit. So maybe I should try a different one. But I mean, you do get the idea, and the carrot is also pigmented itself. Maybe it's 2 or 3 cell layers thick. But now let's have a look at the leaf, which is, where is it? Here. Ah, yeah. Okay, maybe you remember last week or no, 2 weeks ago or so, I also put some leaves under the microscope here. And I talked a little bit about the leaf structure as well. Some of you might remember this. Let's do it like this, okay. Yeah, and I already talked a little bit about the palisade mesophyll and the spongy mesophyll. I'm not going to go too much into the biology part right now, but this here is the top surface of the leaf, and you can see that the cells over here are actually long like this, and the bottom part of the leaf here, the cells are spongy, so there are air spaces in here as well, and that's basically the outer layers here, the epidermis, the upper and lower epidermis, you see, are not green because there are no chloroplasts in there. But maybe this is still a little bit of a very high magnification, yeah. But I think if you just look at this and if you focus through it, you're going to see that actually, there are not so many cell layers. So the cut actually is quite thin already. And the reason why it's difficult to see a little bit is because of the green pigment. And see over here, this is where it's a little bit darker already. So now let's have a look at another one over here. Ah, yeah. Here you can see it quite nicely, the top layer of the leaf with the cells, yeah. And if you actually focus through it, you can see that actually maybe there are only 2 cell layers. There's one cell layer goes out of focus, the other one goes in focus. Yeah. And over here, I think that's the third one. Yeah, this one seems to be a little bit thicker already because it's darker. Yeah. So that is essentially the direct leaf under the microscope. And all of the questions here, what are the little spikes on the sides of those leaves and so on, all of these questions will basically, I'll try to answer later on, okay? Those spikes on the leaves, those plant here are quite common, and especially if you find them on the lower, they have various functions, but especially if you find them on the lower surface of the leaf, then sometimes they are there to actually decrease the loss of water vapor. So over the stomates, sometimes can be found to kind of shield the openings of the leaf to prevent loss of too much water. Okay, just one possible example. Yeah. So those little sticks, okay, I'll not answer the questions now. I'll wait for later on. So, however, I've already done a little bit of other preparation today as well. And what I would like to show you now is what I've done here. These are cuts that I made today in the morning. And look, what color do they have? They are white. Why is that? Because I put those thin leaf cross sections not into water, but into alcohol. And the alcohol removed the chlorophyll. It only took a couple of minutes. The alcohol turned green, and then I kind of replaced the alcohol. And then after a couple of minutes, maybe 10 or 15 minutes or so, they lost all of their color and they turned white. Yeah, and I would like to show you now those under the microscope. So this is pure alcohol. So pure alcohol has the disadvantage that the specimen starts to shrink. Yeah, so and deform a little bit. So when I put them in here, yeah, I can do that. Then there is the danger when it dries out, then it's really going to completely lose its shape and deform. Okay, so that's not something that we want to have. So what I have to do is after placed it on here, I'm just going to put a few of them on here. I'm going to add a little bit of water. Again, I'm searching for my pipette here, and the water will prevent the leaf now from shriveling up. Okay, because if you just want to see how it looks like, just for the fun of it, I'm going to put one of those here now right next to it here, and I'm just going to let it dry out, and you're going to see what happens to it when it starts to dry out. It's not it's actually completely loses its shape. Yeah, so that's why this water is kind of important. I always have to check whether now these are actually 2 cover glasses. Okay, let me try another one here. What did I have over here? Yeah, that's better. Okay. So let's have a look at this here now, simply to show you how a de-stained yeah, leaf looks like. Here, that's the green one, the fresh one, and that's the one, yeah, that is the one without the chlorophyll. Yeah, and you can actually see that it is significantly brighter. And here we're now actually able to see the cells much better. And if you're focusing through it, see it's blurry, 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 blurry. Then some cells come into focus. Just going to show you those little boxes here, just cell walls. Just have a look at those cell walls, right? Yeah. And then let's wait until it goes again out of focus. And so it's out of focus again. Now the next cell layer. It's one cell layer, and well, actually it only appears to be like one cell layer, two, yeah, maybe 1 to 2 cell layers stick only. Yeah, so I think that's okay. Yeah. So this is basically the one that has been treated in alcohol. You see the whole bunch of air bubbles over here, and over here. Ah, look, this seems to be a nicer part over here. Yeah. When it goes into focus and out of focus again, I do not see a second cell layer, you know, going into focus. Yeah. So you see, it's fairly thin. Okay. So if we want, we can look at another one over here. Ah, look, this is actually even thinner. Yeah. And that's the third one over here. So so basically, take-home messages is alcohol, concentrated rubbing alcohol actually can be used to make some of the parts a little bit more visible. Now let's have a look at the one where it did not add any water. Well, okay, of course, without water, it's already difficult. But yeah, you see, it's starting to dry up a little bit, and then it's going to start to shrivel up. And it's also quite dark. Yeah, this has to do something with the refractive index as well. Yeah, so this is basically just something that I wanted to show you. And just because it's kind of fun, let's try the following. Let's add a little bit of methylene blue as well. Let's see if I can do a little bit of staining. I'm just looking at the time. A little bit of staining as well. And I'm going to grab one of those guys here again. And I don't know, whatever looks thin. And I'm going to add a small drop of methylene blue solution, which contains a little bit of alcohol, but also it's not purely concentrated alcohol either. So it will now absorb, hopefully, the stain. And actually, it's designed so that you can actually put a cover glass directly on top of it. Okay, still seems to be quite a thick one. So it asks here how many percent alcohol I used. I think 96% alcohol. It's just the regular, one that as it came directly out. Okay, you do not see that scope view, and I have to find it again. Where is it? Where is it? I can't find it. You see, that's why you should always start at the lower magnification. Nope, that's fibers. You know what? In this case, if you can't find it, you go down with the magnification. Simple as that. Here we go. Okay. And yeah, I don't know if this adds so much value to the whole thing. Yeah. But sometimes the individual cells start to become a little bit more visible. Yeah. Especially down here. Yeah. Yeah. So this, and sometimes you can also see how the stain is actually, it starts to absorb it from the edges first, and as it diffuses into the leaf, slowly the inside part also starts to turn blue. Yeah. Yeah. So. Yeah. So this methylene blue solution is a general purpose stain. Yeah. In this case, I don't know if it really adds so much value because, maybe over here the individual cells that you see better, because Normally, you would like to make different parts of the cells better visible, but if it just stains everything uniformly, then actually there's not, not a lot of point here. Yeah. Yeah. So why does alcohol? Yeah, why does alcohol shrivel up? Because alcohol will actually remove the water. The water will mix with the alcohol. Okay, and then the alcohol will evaporate, so there is essentially nothing left. Yeah? So for fixing specimens, basically fixing means for preparing them. What you do is you sometimes use a mixture of alcohol and acetic acid, because the alcohol causes the specimen to shrivel up, and the acetic acid causes it to swell up again for whatever reason. So you're adding sometimes, alcohol and acetic acid, and this is what is referred to over here. Yeah, it will actually tell you should use FAA. It's a fixative which contains alcohol and acetic acid, and F is formalin or formaldehyde. Gee, one of them is poisonous, the other less so, but still. And this is basically a common fixative that is used for preparing specimens. Okay, so again here, let's have a look here. Yeah. So that's again the view. So you see, a little bit of plant cross sections again. Yeah. So this is something that I wanted to kind of briefly show you. What time is it right now? Okay, it's in around 20 minutes. I'd like to start to talk a little bit about the about the questions that you've asked. Okay, and what I would like to do now is I think I'm just going to slowly start to move over to and show you a little bit the new way of connecting a camera. Okay. Okay. So I'm just reading some of the questions which I will try to answer then, okay? So I will move on, okay? And I want to show you now the adapter, okay, that I have. And I know that this is a very common issue. I need to clean up the desk a little bit. It's a very common issue. So many people want to know about connecting cameras to microscopes, and honestly, there is no simple, easy way, unfortunately. However, there are simply several techniques, and this is simply a new one that I just want to present, which I did not make a video about yet, but a separate video will be made. But I just want to share it with you. And even if you already have a camera connected, and if this is not an issue, it may maybe still be interesting to you, simply so that you know what's out there. And I would like to do a little bit of cleanup first here, because I do not want any of the other camera equipment to become soaked in water. I will also remove this here. Before I do that, look, how do I get it out? Of course, I can turn it, and slowly advance it out. Or a second possibility is kind of just taking some tweezers here and lift it out like this. Okay, so that's the way it goes. And I'm going to go to a hardware shop, and I'm going to get myself a puncher. So let's move this away here, and I just like to show you over here. For the sake of making it a little bit easier, I've actually taken along the head of a microscope here with a photo port, trinocular head, and I would like to explain a little bit. This is from the Swift 380T, but many microscopes have a similar design here. And what I would like to do is I would like to connect this camera here. Okay, so it's a DSLR camera. I would like to connect it over here to this photo tube, and the question is why. Okay, those cameras have a couple of advantages, a whole bunch of disadvantages as well. But one of the advantages is that they you can do movies, quite videos. That's USB cameras are a little bit slow to do that. The image quality is quite high. The sensor is large, right? And some people simply might already have one of those at home. The only question is how do I connect it, right? And I made already a whole bunch of videos, but the most common way, the one that I always showed you up to this point, is that you connect an adapter over here. This adapter looks like this. that there is a general adapter over here, some optics in here, and this here is a so-called a T2 adapter ring, and this one is camera specific or company specific. Okay, so this one is for Canon, and then what you do is you connect it like this, just like a regular lens. It locks in place, and then what you can do is you can insert it directly over here instead of an eyepiece, hoping that the microscope doesn't tilt over. So it's not a recommended way to connect it here. It works, but it's not recommended. Or you connect it here on top. Okay, so that is the traditional way. Okay, so yeah, straightforward. What are the disadvantages? It's a pretty huge thing, right? That's disadvantage number one. It could be more compact. Disadvantage number two, not a big one, but still, is that there are optics in between, and any optics that you have in between actually degrade the image quality to a certain extent. Okay, internal reflections and so on. And disadvantage number three is this adapter here. Is sometimes also not that cheap, not super expensive, but it's not the cheapest solution. So it's, I don't know, 150 euros or dollars, and then you have to get the camera-specific T2 adapter ring as well, okay? Not counting the camera itself, obviously, right? So that is essentially the method that's been used quite commonly, and I talked quite a bit about this. And then recently I discovered this thing here. I cannot believe it. And this thing works under one condition, and that's I would say, the biggest disadvantage. Because, and you have to check, because some of the photo tubes allow you to disconnect the top part here. Okay? And what you got here, this is a so-called a C-mount. And a C-mount allows you to attach USB cameras, also industrial cameras, and now here it comes without optics. Okay, without intermediate optics. So basically the image is created by the objective of the microscope and is directly projected into the sensor. Yes, and so the distance has to be a little bit smaller. And what I have here is I have now a C-mount to Canon EOS adapter, and you see that basically this one here will screw in place here. Okay. Now I cannot show you the camera view here because, yeah. And then the camera goes directly on top here. I will also show you how this works. It goes directly on top here. It locks into place just like a regular lens. And then that's it. Okay? And what will happen now is that the image is directly projected into the sensor. And it kind of works. I say kind of because there are a few disadvantages. And the thing is the following. You have to adjust the length of the tube in such a way that the image is in focus in the camera and also through the eyepieces, or at least more or less in focus. You don't want to turn the focus knob too much to find the focus. And it kind of is in focus, kind of, okay? I was a little bit off with the 4x objective, but it was a slightly blurry. I had to turn it a little bit to get it into focus, but that was fine. Okay, with the higher magnifications, it worked fine. And over here, there is a possibility to, and I have to disconnect this again to show you. Okay, there is a possibility to adjust the length. Let me put the alcohol here away. Yeah, there is a possibility here to adjust the length by rotating it out, and then by turning the other one, you kind of can fix it again into place. Yeah. So this is essentially something that I recommend that before you start investing something into here, maybe you just want to try out one of those cheaper C-mount to DSLR adapter rings. Yeah. And I've seen on Amazon, I mean, how much did I pay for this? Around 10 euros or so. Yeah. Considering the fact that it's only a piece of metal, it's still, yeah. But it's at least, if you already have a DSLR camera, at least significantly cheaper than those here, and you might be satisfied with this. You have to try it out, okay? But again, I need to say this only works if you actually have a photo port that allows you to directly connect a C-mounted camera, okay? And you got to check because you might not know that the top part here can actually be removed. Okay, so this is just something, yeah, that I want to, yeah. Would this only work for trinocular scope? Essentially, yes, because you cannot connect this here to the eyepieces. Yeah, and it's not recommended anyway that you connect the DSLR here because the microscope might tip over and you might need additional support. Okay, yeah. So this is something I just wanted to share with you because it is something that I actually didn't know that it existed, and I just recently discovered it. I tried it out, and it actually works. There is a little bit, depending on the objective that you use, there might be a little bit of vignetting. So this basically means some of the corners might actually be a little bit, how do you call this, darker. But the field of view that you're getting is quite large, and that's also quite nice. So yeah, before trying other things, I would try this here. Okay, so that's basically number 2 that I wanted to show you. And I will make, in my other YouTube channel, I will make a more detailed review of this, but simply because some people are really interested in this, I decided to show it to you anyway today a little bit, and maybe some of you might have questions, and I'm able to, answer this as well. So let's put this again away. Okay, and what time is it? What time is it? Okay, still have around maybe a short quarter of an hour, and I would like to try another thing here, and this is the following. What I prepared, because some of you were again asking a little bit about cyanobacteria, and maybe you've watched my 2 cyanobacteria videos in this channel. And what I've done today is today in the morning I added some of those samples with containing cyanobacteria on the slide, and I allowed it to dry. So let's have a look at this, how it looks like under the microscope. So these are now dry. So it's basically I'm moving on to the next topic quite fluently, and I wanted to show you how those look dried. Where's the scope view again? And one of the issues or problems with dried samples. Okay, and it looks kind of messy. Okay, so what you see here, these are cyanobacteria, and it's just switch on D. I see no cover glass now. Yeah. And what you see is because water also contains some minerals and salts. You can also see that all of those little dots that you see here are basically also calcium carbonate deposits and salt deposits and so on. It almost looks a little bit like crystalline over here, like crystals forming. Okay, so this is basically without mounting medium. And what I would like to try to do now, let's go up with the magnification, right? And let's see how this looks like. Yeah. And I would like to see now, how does the appearance differ when I add some mounting medium over it? Because sometimes folks want to know, okay, why is the mounting medium so important? I said, well, it's not only for preserving the specimen and protecting the specimen, but also for actually improving the image quality. So and the question is now is which mounting medium do I use? Okay, I mean, there's so many different ones here. And generally speaking, and that's maybe one of the things that one has to consider, is generally for water specimens like these, delicate water specimens, algae, cyanobacteria, and so on, you generally should use a mounting medium called glycerin gelatin, which is a semi-solid mounting medium. The only problem with that thing is it's horribly difficult to use. You have to warm it up to 45 degrees centigrade because it's solid. When it's at room temperature, you have to warm it up carefully. If you warm it up too much, it's going to break. Break means that it won't become solid anymore, because glycerin, the gelatin will actually break if the temperature is too high. So it's not ideal. So what I'm going to do is I've used have you guessed, I'm going to use another water-based mounting medium, the glue one. Okay, and see, whoa. Can you believe this? That's the bad news. See, something unexpected. That's usually always a demonstration effect. But I did not expect this to actually be completely... Okay, here we go. Yeah, you have to be careful if there's some glue on here. It, of course, glues everything. So let's add a little bit of this water-based mounting medium, the glue, on here. And this glue, because it's based on water, will probably also start to rehydrate the cells a little bit. The cells will start to absorb again some of the water, and I hope that this will also improve a little bit the overall image quality. I do see a little bit of a small air bubble here as well. So maybe I'm going to use my tweezers here. Yeah. And let's drop this on here, and I simply wanted to illustrate a little bit, yeah, on how this looks like. If you do this, be super careful. If there is too much mounting medium on the side, then the objective might become contaminated with the mounting medium, so be really careful. You only use it as long as it's still wet, only the low power. But I just wanted to show it to you anyway. Scope view, how does this look like? I have to find again some... Yeah, and yeah, bubbles, maybe, yeah, but ignore the bubbles. But I think you do see that essentially the image quality overall looks much better, and especially in the background, you don't see these dots anymore and so on. So why is that? The reason is because the difference in refractive index to the mounting medium is not as big anymore, right? And for this reason, the image simply looks much cleaner. Okay, yeah, so that's an issue maybe, but we're going to see how the bubbles behave when actually when it starts to dry up. Yeah? But I just wanted to illustrate this to you a little bit. I would just like to illustrate this a little bit to you to show you the importance of mounting medium. Yeah? So I do see that there are already quite a few questions here. Okay, please do keep on posting, and in a couple of minutes, maybe in about 9 minutes or so, I'm going to go through. The questions. Okay. Yeah. So be careful with But then again, of course, when objects dry up, then they will lose their natural shape, and there is usually you might be introducing some artifacts, and it still looks different. It still looks different than actually if you were to take a fresh sample. Okay, but still, I think you do get the idea a little bit that this seems to be working as well. Okay, you just take a small sample, allow it to dry, and one of the nice things that I see here is that. Let's remove the, you see that they're green as well. This is not bright field, kind of showing the photosynthetic pigment of these cyanobacteria. This is kind of the thing I just wanted to show you a little bit here. Okay, so let's do the following. I do have another one over here. Let me have a look at the next one. What is this here? This seems to be some other algae. Okay, this is now way overexposed. Let's go down a little bit. These are now some true filamentous algae. Okay, all dried up now. Okay, there were a whole bunch of rotifers in here as well. And when they lose water, then they completely shrivel up. There seem to be some tiny diatoms maybe over here. Let's have a look how this actually now looks like once you add some mounting medium as well. Yeah, these are diatoms. Up here, and we do the same thing. We add a little bit of, where is this, the mounting medium. The water inside the mounting medium again will rehydrate some of the cells. And I'm going to try to find again a cover glass. Here we go. Yeah. And let's have a look here. And let's put it on here. Allow capillary action to spread, to do its work so that the glue spreads And let's put it again under the scope. Wrong scope. And here they are. Yeah, it's a little bit better maybe. Those, by the way, those dark things that you see here, these are air bubbles which appear to be apparently inside the cells. And the air bubbles are quite visible because of the big difference in refractive index to the surrounding medium. Yeah, so maybe it would be a good idea not to allow the algae to dry out completely, but maybe they should still remain moist. So no excess water, of course, but maybe they should remain moist when you add the mounting medium. Yeah. So you know what I'm going to do now in a couple of minutes. So maybe we can slowly start to finish up posting questions, or maybe if you were interested, maybe you can start to post the remaining questions, because in about 5 minutes or so, or 4 minutes, I'll be starting to answer some of the questions. Of course, you can also post questions later on, but I'll get a sheet of paper with all the questions printed on, so that you're also able to follow it then on my table. Yeah? And this is something that I would like to try out, then basically I hope that this is going to be a little bit more organized. Okay. By the way, a couple of other things. If you have any specific wishes of things that you want me to put under the microscope for future live streams, please also do comment on that and write comments. I'm always grateful for any help or advice or suggestions. Okay, because I do have to sometimes think up a little bit the topics. I know that sometimes it can be a little bit repetitive of what I'm doing, always showing algae again and so on. But sometimes there are always new people joining into the live stream who have not seen the previous ones yet. But if you have any other specific original ideas, then please feel free to suggest them. Okay. Here you can actually see that they look a little bit larger now, the cells. Because they started to swell again as they reabsorbed some of the moisture from the glue. So, I'm now waiting. I'm currently waiting for the printout, because I do receive some help today. ChatGPT will be able to suggest new, unique ideas. As a matter of fact, you're going to laugh. I actually did ask ChatGPT for possible video ideas. Some of them are quite interesting, but honestly, sometimes a little bit difficult to do. And sometimes it just is stuff that I have been doing already before, because I've been already covering quite a few different topics. So I'm basically now waiting for the printout, because I do receive some help today. But maybe I'm able to see something interesting here as well. Of course, one of the disadvantages of permanent mounts, of course, is always that you do not see any movement. That's of course always a little bit of a pity. But I did receive now. Look what I got here. Whole bunch of questions. First of all, thank you very much. And look, here are your questions. At least the ones that I got on time. If you have more questions, please do post them. But these are the ones that I got. And the people who have also posted them, questions and comments. Okay. And some of them, I hope that I'm able to answer them. I'm looking at a second-hand sliding microscope or microtome. Are these just for meteorology, or are there other uses? I'm a little bit confused right now, because they are so-called sliding microtomes, and meteorology. I have to admit, sorry, I'm not able to answer this question, because you have to be a little more specific. I don't fully understand what you're specifically looking for. Okay. So what are some little spikes the size of those leaves? Okay, those spikes here that I showed you before, those spikes or those many leaves have those leaf here on some of them can be quite fancy, called trichomes, and they can serve many different functions. Of course, they can serve as protection. For example, certain insects, they don't like them. Right. However, what I mentioned is that if you find some of those here on the bottom side of the leaf, you also have those stomates, the openings. And through those openings, the leaf receives gases. Carbon dioxide, for example, from the air. It releases oxygen gas, but also over those stomates, a lot of water vapor is lost. So there's a real danger of the leaf or the plant drying out. And having a lot of these fuzzy little tiny hairs covering the stomates, this reduces the loss of water vapor because it kind of blocks it a little bit. So gases like oxygen and carbon dioxide, they can go in and out quite easily, but the water is kind of caught in there. The water vapor is caught in there. So, but ultimately those hair on leaves can have many different functions. Some time ago, I made actually some videos about the spikes of a stinging nettle, and they basically contain substances that causes your skin to burn. So you can see it's also a defense mechanism. So short answer, many different answers for those little hair. Yeah. Can you embed the specimen in paraffin prior to sectioning? The answer is yes. However, and that's the thing, those sectionings that I've made with a carrot, right? If you want to embed this in paraffin, it takes quite a bit of time. And it works like this. You have to carefully first dehydrate it. That means you have to put it into alcohol, but you cannot put it into alcohol directly, but you have to slowly increase the concentration of the alcohol. Otherwise, there's going to be too much shrinkage. And you have to leave it in each alcohol concentration for at least a day or so. Then you have to add a substance. Traditionally, a substance called xylene was used, which is not healthy. You do not want to use this in your household, but especially in hospitals, they are now using alternatives for this, okay, which are less harmful. And then you have to embed it in paraffin as well. Paraffin is wax. And then you have to slice it. You have to section it. Then you have to use again xylene or other solvents to remove the wax again, the paraffin again. So you see, it's a fairly lengthy process to do that. The question is always, is it worth it? And it is worth it if you want to, for example, do this commercially, for example. But if you just want to observe for hobby purposes, some things, unless you like making paraffin embedded specimens, for hobby purposes, it just basically is fine if you just use those microtomes that I showed you to make direct cuts, even if they might not be quite as perfect. Okay. So do you think it's possible to 3D print an adapter for DSLR camera with a Swift? So basically, short answer is yes, it would be possible. However, you'd have to know all of the dimensions and everything. So for example, the Swift 380T has this, at least my version has this C-mount. Okay, so essentially you could think about 3D printing this here. However, I mean, I don't know how, I don't know, there are overhangs here, and I don't know if this is going to be so easy, right? And considering the fact that this is fairly cheap, yeah. And another thing that I wanted to say is that the distances have to be correct as well. And I don't know if such a thin 3D printed adapter is actually going to be very stable. Okay, so this is a little bit a question that I have. Yeah, so I have already 3D printed adapters. Also made a video about this for another microscope. Yeah, but it depends really a little bit on the folder tube that you have. Yeah. More of a biology question, but why does the alcohol shrivel up the specimens because of osmosis? Yes, alcohol, at least ethyl alcohol, is very water soluble. So basically what will happen is that the water from inside the cells will actually go into the alcohol, right? If this happens too quickly, then of course the cell loses a lot of water. Okay, and then now if you take it out and then the alcohol evaporates, then there is no liquid anymore, and then the whole thing starts to shrivel up yet more. Yeah. And as I already mentioned, in microscopy, if you want to prevent the shrinking, they found out that you can add acetic acid, which is kind of the acid found in vinegar, right? And because this has a compensating action and causes the cells to swell again. So there are actually recipes on the correct mixture. Please talk again about the little sticks growing on the side. Oh, okay, I mentioned those. If regular, there are stomata protectors, and if irregular cyanobacteria, as discussed elsewhere, okay, not cyanobacteria. So those over here, if you look, I don't know if you're able to see this, not cyanobacteria, because cyanobacteria are actually growing in water. And if you watch the video that I showed you, I also uploaded one today, you could actually see on the surface of some algae, you could actually see cyanobacteria growing. However, the object was underwater. Yeah, so that means they were kind of growing on the leaf, under the water. But this one obviously is from a flower, it's not under the water. And I don't know if the stereo microscope is able to show this, because I don't know, because we're not seeing it from the top. Let's see. I mean, here you see some of these here on the side. You always have to check the edges, right? Yeah, and you see over here, that there are these tiny little protrusions here. I don't know what function these ones specifically serve, but I know that some plants actually have a lot of them growing right around the stomates to actually reduce the loss of water vapor. So, but here under the stereo microscope, you're able to see them here as quite well as well. Okay. So let me move on quickly. Where is desk view? So again, the cyanobacteria, these are basically separate cells, and they grow on other plants, for example, or on rock surfaces underwater. I seem to remember that methylene blue attaches DNA, that's correct, thereby making the nucleus in the cell visible. Do I remember this correctly? Yes. And the thing is the following, the staining that I've just done right now did not, I've made some stains before where you could actually see the nucleus quite well. In this case, not. It could be, and again, that if the cut is too thin, right, then what might happen is that you're cutting open the cells, and then all of the cell's contents are lost, including the nucleus. Yeah? So this means that if you do not see a nucleus, it does not mean that there is none there, but maybe it was simply lost during the sectioning process as well. Another thing, the methylene blue solution that I'm using here is called Löffler's solution. It's a ready-made mix. Löffler was a German biologist. I'm looking for a pen right now. He basically, this is not a concentrated methylene blue solution. And this one contains also alcohol. And it contains also a base, I think potassium hydroxide, if I remember correctly. And because it's alkaline, the stain, it likes to stain acidic parts of the cell. And what's one acidic part of a cell? That's the DNA. Because DNA is an acid deoxyribonucleic acid. So bases like to stain acids. So that's also one of the reasons. And then maybe methylene blue itself is also slightly basic, but the solution is as well. That's why it stains the nuclei especially well. Yeah. Would this only work for a trinocular scope? Connecting the camera with this one here, yes. As I already mentioned, because only a trinocular scope, and not all of them, only trinocular scopes actually have a C-mount adapter. Yeah, so that is the important thing here. And those C-mount adapters, they do not require intermediate optics. Yeah. How about use a 0.5 magnification tube? About those magnifications, that is basically referring to this one over here. Okay, it says here that there is a 2 times magnification. And sometimes you have a 0.3 times and a 0.5 times. Now, why? Why is this? The thing is the following. And this is again goes a little bit into has to do with the size of the sensor, because the sensor of a digital reflex camera, the DSLR, this one over here, let me get it again. I just want to show it to you. I mean, many of you have already seen this, but just to illustrate this, yeah. I mean, there's a mirror in here, right? And if I am to open this up, I have to, let me quickly, okay, I need to, let me quickly do this. There's no card in camera. Yeah, I mean, if you press this, yeah, great. Just a second. Let me quickly, I don't know if it's even going to release because I don't have a card now. Okay, it doesn't matter. In any case, you get the idea that the sensor covers nearly the whole area here, right? And so this basically means that the sensor over here is significantly larger, than the diameter of the tube. Okay, and for this reason, you need a magnification which is greater than 1. So to essentially take a small image and to make it larger. Right? And if you have now a small USB cameras with a small sensor, then you have to make the image, actually you have to compress it down to make it fit on the sensor, right? And that's why you have magnifications of 0.3 or 0.5. So the magnification over here is important for adjusting the size of the image to the sensor size, and each sensor size has somewhat of its ideal magnification. So this the idea of this magnification is really not to change the magnification of the image, even though it also does that. But it's actually there to adjust the size of the image so that it fits on the sensor. Okay, so that's kind of the idea. If this magnification is way too much, then essentially you're magnifying it much more than necessary, and then you're only taking out this with a small sensor, a small central portion, which is also not what you want. So this kind of the idea is that the magnification over here is actually there to adjust the image to the size of the sensor. So if you're asking here about a 0.5 magnification tube, then this is actually used for cameras, USB cameras that have a small sensor. Thanks for the live. Small question: how did you do the polarizing light in the Swift 380? Not only Swift 380, but pretty much any. Any one. Pretty easy. I made videos as well. I don't have polarizing. You take 2 polarizing filters. Just a second. Give me 10 seconds. Yeah, you buy those polarizing filters. There's now a protective foil on here, right, and which you want to remove. These are linear polarizing filters. They don't cost so much. You can cut them with scissors. And what you do is you buy one of these things and you cut it apart. And then what you do is you put one beneath the slide. Okay, you take the other one filter above the slide and you rotate it. Okay. And then you have the objective on top. And that's the easiest way to do polarization work. And if you use a small magnification only, then there is enough space. So basically what you'll have is, I'll use a different one, one where there's no mounting medium on it. So basically you'll take without the protective film, okay? You put one on top here, and then you have the microscope objective on, directly over here. Low power magnification, and it's not a problem. With a high power, you might be actually touching the foil, and you don't want that. So, do you thin the PVA glue with water? Yes, that's correct. I used some PVA glue and I added some water. I did not even measure out how much water. It should kind of flow easily. So maybe, I don't know, 10%, 20% water. Right, and if it flows easily, then essentially, it's fine. The water itself takes some time to completely dissolve. So I had to wait, I think, overnight and shake it gently to mix it in. Would using water-based mounting media make bacteria grow? No, definitely not. Because the point of mounting media, a mounting medium, is actually to dry up and to preserve the specimen. Okay, so that's actually the point of a mounting medium is exactly the opposite of this question here. You do not want the bacteria to grow because if there are any growth or changes happening, then that defeats the purpose of the mounting medium. So for example, I don't have a slide here right now, but let's say that let's just do the following. Let's say that I've just made this one over here, and let's say that for whatever reason bacteria were to grow in here. Then what's going to happen is they're going to decompose the specimen. So that's actually something bad. You don't want that. And when you have decomposition happening, decomposition is a fancy word for rotting. Okay, if you have the specimen rotting away because bacteria and also fungi break it down, then sometimes you also have decomposition gases forming, for example, carbon dioxide gas or even methane gas. And this actually can happen. And all of a sudden, after some time, you're actually going to see bubbles forming directly around inside the specimen. And the reason is because bacteria have become active and they started to produce those gases. Yeah. Decomposing the specimen. And for this reason, you do not want to have bacterial growth if you want to make a permanently mounted slide. Now, I know that sometimes you actually want to observe the growth of bacteria. And in this case, of course, you need a nutrient medium so that the conditions are such that the bacteria actually wants to divide. But then, of course, you do not have a permanently mounted slide. Okay? So even water-based mounting media actually by the way, glycerin gelatin, which I mentioned before, which contains gelatin, which is actually a protein, which can be used as a food source, actually has a problem that if it's not properly done, then actually might also, fungi and bacteria might grow. And for this reason, they actually added sometimes disinfectants to the mounting medium, to the glycerol gelatin, so that actually it will block the growth of bacteria and fungi. Yeah, so that's simply something that I wanted to make because it's an important topic that essentially you do not have bacterial growth. I have issues with this the Elmer's PVA. After a few days, it dries under the cover glass. Well, that's what it's supposed to do. Well, actually, that's what it's supposed to do. Okay, the idea of using this glue is actually to make permanently mounted slides. Okay. Basically, the reason why I like this medium is because it is non-toxic. I mean, it's even child-friendly. And for those of you who don't know it, just a second. Again, I have to reach over into my cupboard. That's this glue here, right? It says child-friendly. And it's the clear version, right? And the idea is to essentially make slides that last for a long time. Yeah? With the disadvantage that you do not see movement anymore, obviously, right? But at least you have a slide that you can put into a slide box and hopefully you're able to store this for a long time. Yeah? And many mounting media essentially are toxic. They contain solvents that are not good, or the specimen has to be completely free of water. And when I say completely free, I really mean completely free. It has to be not only do you have to remove the water with alcohol, then the alcohol it has to be removed again, and it has to be completely water-free. That's why they've used xylene and other solvents. And that's not something that you want to do at home, right? Because the fumes and so on. So I was kind of looking around for a cheap mounting medium for hobby purposes. That's it, okay? I'm saying for hobby purposes because if you really want to do this for research, then you have to worry about the refractive index of the mounting medium, a whole bunch of other parameters as well. But that's something that we hobbyists really don't have to worry about so much, right? And that's the reason why I use this here. And you can also mix it with glycerin. So this actually does work as well. Depending on the amount of glycerol that you add or glycerin that you add, it stays kind of semi-solid, right? So the glycerin will actually also attract water, moisture from the air, therefore I can imagine, especially for slightly more delicate specimens that do not like to be completely dried, some of the glycerin can be used, also because the glycerin can be used also to adjust the refractive index of the mounting medium, if this is actually important for you, okay? So that's something as well. If you want to get rid of the air bubbles, put 1 drop of alcohol on the slide and the cover slide, it will break the surface tension, so no air bubbles. That's correct. As a matter of fact, sometimes if you take, it's kind of interesting, if you put a specimen that is kind of moist and then you add some alcohol on top of it, all of a sudden you're under the microscope able to see air bubbles forming. Right? Because apparently it's not only the surface tension, that's one thing. the alcohol will break the surface tension. But I think a second factor might be that maybe the dissolved gases that can be found in water cannot be kept anymore if you add alcohol. This kind of pushes out the gases, and that's why you have bubbles forming. Yeah? So you can actually sometimes see this even under the microscope, that all of a sudden you add water and alcohol, and then you see bubbles forming all over the place, kind of pulling out the water. However, there is also, I think somebody also mentioned this, is one way of what you can do is if you have mounting medium like this, you can also try to apply a vacuum, because it will also draw some of the dissolved air and the gases out of the liquid. So I some months ago also made a video of this. Some people actually also will place this slide into a vacuum chamber, and there are even homemade vacuum chambers, right? So you basically will force the air bubbles out. So what I would probably suggest is the following, that you maybe force the air bubbles out maybe before you apply the cover glass. That could also be a possibility. Yeah? But indeed, there are several ways how you can deal with air bubbles. And I'm going to wait now until next week, and we can have a look. I don't even know where I put the slide. We can have a look the next week again how well this, that's still on here, how well this actually will have worked, and if there's still some bubbles present. Okay, let me quickly, where is the scope view? Yeah. Here you see that, there's still some bubbles here present, and let's see how this actually behaves when the glue starts to dry. Okay. Yeah. So these were all of the questions here, but maybe I'm quickly going to go through. Maybe some of you have also posted some more questions. Yeah. So I got this here. ChatGPT. Did you ever make a Feuchtekammer? The answer is yes. I need to explain what this is. Basically, what you do is if you want to, I don't know, let's say you're taking, I've got some jars over here with some water samples, and you put a water sample and you make a slide. And then the question is now, okay, you put a water sample on here, cover glass on top, no permanent mount, right? And you see the little water microorganisms moving around, and you kind of want to keep the slide now for a day, 2 days, a week maybe. The problem is it's going to dry up, right? And the problem then is everything's going to be dead, right? But you don't want that. So what you do is you take this slide and you put it into a plastic box which contains water. Yeah. So I don't have one here now, but I actually you make in a plastic Tupperware box. You make some kind of a rack where you can place the slide, and then on the bottom you have water. So the moisture is 100%, and this will actually prevent the slide from drying out. So this is it's the German term. It's called Feuchtekammer. It's a wet chamber. Yeah. So that's the thing. Yeah. So first time I heard the word microtome. Yeah. Well. Okay. I took one drop from a greasy dish filled with tap water under my microscope, and I found strange creatures. Yes. I've sent an email about ideas. Could you just read it? Yes. I have to check where the email is. Okay. I sometimes receive so many emails, but thank you again. I will have a look. Maybe it ended up in the spam. Okay. Can you show us cannabis flower slide? Actually, the thing is the following. I don't have cannabis. However, I know that there are possibilities where essentially they are growing those cannabis plants, not for the use of drugs, obviously, but because as a cultural plant, because the fibers, the cannabis fibers, also have a use. Yeah? But honestly, I will tell you the cannabis plants will look pretty much the same as other plants as well under the microscope. You've got cells, you've got stomates, you have all these things. Yeah, it dries and disappears, evaporates. I guess you mean the wet chamber. Okay, and would like some live streams with more water sample experiments, like where you squished the Iolosoma air bubble or tested how paramecium responds to electricity. Okay, thank you very much for the recommendation. It's been quite some time when I've done that. Several months ago, maybe a year ago or so, or even 2 years ago, I actually applied an electrical current to paramecia, and you could actually see that they were kind of moving, towards the different poles. Yeah. So this is something, I still have the power supply, obviously, so I can do that. I have to grow, again, a few paramecia from the water sample that I have. Yeah. So that is, I meant the PVA glue evaporate after days while drying. It disappears. The thing is the following. Let's put it this way. The PVA glue contains, of course, water. And when the water evaporates, then there is always a certain amount of shrinkage happening. Now, it could be that maybe if you added maybe too much water to the PVA glue, then the water will evaporate, and then there is actually too little of the actual glue left on the slide. So maybe this could be a reason. Okay, so if there's too much water in here, it's nice because it flows very nicely, but then there might be also more shrinkage, and then it will actually become smaller. Yeah, so this could be a reason. I think I understand now what you mean. Yeah. That it starts to disappear, and then you have actually bubbles forming. You see, you have to kind of find a little bit the optimum spot. Not too much water added and not too little. But I found out that the glue, as it comes out of the bottle, is also very thick. It doesn't flow well, right? And it also becomes more difficult to actually remove the air bubbles. Okay, yeah. So. Have you ever tried using ultraviolet light to see if bacteria react? Well, I can tell you the following. Not under the microscope, but I've treated some water samples with ultraviolet light, and yes, it kills bacteria. So I think the important point here is the following. If you add certain substances, chemical substances, or even ultraviolet light, and if you treat, let's say, bacteria with them, this does not make the bacteria disappear. The cells are still there, they're just dead, but they're still visible under the microscope. So this basically means that just if you see bacteria or cells under the microscope, this does not tell you anything on whether they're alive or not, or at least not always. I mean, certain bacteria are able to move, so you know that you're alive, but not all of them do that. Yeah? So that's the reason why when you're in microbiology, when you actually want to determine how many live bacteria are there in a certain sample, you do not do that with a microscope, because under the microscope you cannot distinguish live from dead bacteria that easily. You need to actually grow the bacteria on a petri dish. Yeah, so this is a little bit a thing here. So while bacteria will respond to ultraviolet light because UV light will actually damage the DNA, it will kill the bacteria, or at least many of them, you do not see this process under the microscope. Yeah, so that's a little bit the limits of microscopy. Yeah. Could you use super glue on the edges of the cover glass to prevent the PVA glue to dry out and evaporate? Yes, you can do that. As a matter of fact, the super glue will react with the moisture of the water. However, you have to be aware that when you mix water with super glue, and you can actually also see this under the microscope, sometimes there is some clouding happening. So it turns fuzzy and so on. So you have to be a little bit aware that the fumes of the super glue will kind of deposit itself. So be careful. Do not place a slide with fresh super glue under the microscope, because the fumes of the super glue will actually deposit also on the lens. The fumes will deposit also on the lens of the microscope. So be careful of this, right? So if you want to experiment with super glue, just make sure it's completely solid and not liquid anymore before you actually place it on the stage of the microscope. Thank you. I like this new format, but who printed this sheet? Haha. My son did. Okay, he copy-pasted. Thank you, by the way. To him, he copy-pasted the comments. I asked him to help me here, and it also makes it easier for me, and it also makes it easier for you because you're able to read it. Okay. Yeah, don't forget to press the like button. Yes, always appreciate it. Yeah, and I think for this today, I'm going to leave it again. I will basically, yeah, now that I'm back from the holidays, continue to make a couple of videos again. Please do also check out my second channel. The second channel is Microbehunter Microscopy. There is a link included, and this second channel is more less on observation, but more on microscope optics, microscope reviews, and so on. I decided to make a separate channel for that because it sometimes does not fit well into the observing part as well. So I'm going to simply leave it at that. I hope to see you again in the future. If I always do the live stream usually on Saturday. If for whatever reason it's not possible for me to do it on a Saturday, please check the announcement. And I sometimes also post community posts to tell you if I have to move the live stream. So enough for today. 1.5 hours is long enough. It's already quite late in the evening where I am right now. I wish you all the best. Happy microbe hunting as always. See you around next time, and bye-bye. Bye-bye.